Good. Then, good morning, everybody. Thank you all for coming. Today it's the last seminar of the year from our cluster seminar. And yeah, today I'm very excited to announce Professor Andreas Thum from Leipzig. Yeah, he's not a stranger to Konstanz, so maybe a little bit about his CV. So he did his PhD in Würzburg with Professor Martin Heisenberg, back then in the fly, I guess, adult fly, no? Yeah. Learning and memory. And then he moved to Switzerland to the lab of Reinhard Stocker, where he started working with the fly larva. And he got an Eminöter grant and then moved to Konstanz to start his own lab in 2011. Yeah, 2011.
If you say that.
Yeah, I think something like this. So he was here for 6 years, also a member of the Zukunftskolleg, and then I think 2017 he became a professor in Leipzig. Yes, exactly. Konstanz let him go. And he's now professor in Leipzig at the university in the genetic department. And also I just learned that he recently got promoted to director of the Biology Institute.
Promoted,
yes. Okay, yeah, and then thank you for coming and looking forward to your talk.
Thanks a lot for the invitation. And yeah, it's somehow special to come back, or it's like coming home somehow, but the home looks a little bit different and parts exist that didn't exist at the time when I left. Yeah, so I will talk a bit about the connectome and other stuff, and not about genetics. That was what I heard before, not too much about genetics. Yeah, so usually when I start these talks, the intro is somehow like that the idea or the focus of the lab is that we want to understand how the brain works, something about the function, and then to know how a brain works and how neurons communicate and how the interaction works, we have to know the structure. So what are these neurons, how do they talk to each other, and so on. So that is the idea. And therefore, can you convince a little bit, or maybe you know already, and that it's like that. I think that the Drosophila larva has several advantages to work with the larvae, to look at that. Of course, also disadvantages, and I will not talk about the disadvantages, of course, today, but only about the advantages. Okay, and of course, something like that, the approach or the main advantage of the larva is its simplicity. But for years I told everybody it's super simple, but then I looked at it and realized, no, it's not so simple, but we can handle it. That's the important thing. And so you need a system. I mean, that is what Kandel said in his Nobel Prize lecture. You need a system that is not super complex. You first need a system that is simple. And there we look at memory storage, but you can exchange that. You can just look at information flow in a network. And then we want to study that and how that affects the behavior of the animal. And of course, in an organism that we can handle, that we can manipulate, we can come up with an hypothesis, and then look at the circuit and the behavior output, what is happening. Okay, so here I'm in collective behavior, so is the topic, of course. And I stole this from your homepage. So I didn't Google myself, but Googled you. So and what you say is on your homepage is that your research seeks to quantitatively measure the collective behavior by considering the actions, traits, and states of individuals, the structures they form, and in which they interact, as well as the environmental issues they are subjected to and create. And if you translate that somehow to what we are doing in the lab, so maybe it's possible to say, okay, the individuals we are looking at are individual neurons. And the structure we are looking at, this is the neuropil, so the sub-organization or the substructures or structures in the brain, like for example, here is the mushroom body, the learning and memory center, or the integration one of these integration centers, but there are many other subunits. So you see a certain structure. And of course, the environment in that all the communication happens and the interaction is the brain. And this is just taken from the publication 2 weeks ago. So now with the Drosophila larvae, based on the approach of Albert Cardona and 100 other people, it was possible to reconstruct all the connectivity, all the individual neurons, and all synapses in one brain. And now we have the information. So what it means, and this is also stolen from your homepage, so we are interested in, or now the field, when I started to work 10, 15 years ago, the main interest was on particular neurons and what is the function of individual neurons in a network. But now it's obvious that a brain works in a different way. It's an information flow through different pathways, and we are interested in analyzing the information flow in these networks, individual neurons, but in principle, I think for some of these approaches, it doesn't matter if these are individual neurons, if it's fish, if it's some communication happening in social networks. So the way how the information flows and so on, that is important and we are interested in. And our network is the brain of the Drosophila larva, which consists of about 10,000 neurons. And however, as I said at the beginning, I usually said it's very simple, but it's not, because we have much more than 1 million neurons, synapses. And that's too complicated to remember all the connections. But it's possible to handle that with the computer science or with the computer methods that we have. Okay, so just coming back to the animal, that's the larvae, as most of you of course know, and the network, the brain, is here located with the two hemispheres in the front third, and it's similar to the humans. It's connected to the external, to the peripheral nervous system, getting the information here from the head. There are a lot of sensory systems located at the head, as we have with our heads. And you also see the connections with the nerves to the muscles, to the abdominal segments, and so on, similar as in the spinal cord of humans. So there are some things that you can compare, but of course you have to be careful on a different level. Okay, so the outline for the talk after this intro is that I will tell you a little bit about the connectome, where we are right now and what is the next step we are doing right now, we are trying right now. I will then talk about the larval dopaminergic system, a collaboration also with Katrin and others. And then finally, I will tell you something different. Okay. So, okay. So, I mean, all this approach, and this is now happening at multiple levels and in different animals and so on. But that started more than 10 years ago with Albert Cardona and also supported by Jim Truman at Janelia and, of course, the entire infrastructure in Janelia. And the idea was to reconstruct the adult brain, which they are now doing. But at that time, they started, just a head start had the Drosophila larva. And the idea was more or less to test if that is working. And it worked that good that from that an independent project developed with a visitor program, and more than 100 people teamed up to reconstruct it. Why 100 people? Because one thing is you have to create a volume where you cut the brain into thousands of sections, and then you scan them with an electron microscope. There are different techniques, and it's improving and so on. And then you have to put the data together in 2D and later on then in 3D, and then you can do segmentations in these 3D volumes. So the volume for the larvae looks like that. You see the circles here are the cell bodies, and in between here's where in the neuropils here are the neurons talking to each other. These are the two brain hemispheres, and here is the ventral nerve cord. And you always see at the outer layer the cell bodies localized, and they exclusively project into this area where they talk to each other. And then you can zoom in, of course, and you can follow through the Z dimension, you can follow neuron by neuron, and of course you see stuff like mitochondria, other things if you zoom in more. But you also see these structures here, particular these T bars. These are specific for Drosophila, what you see here. And these are the presynapses. And if you look a little bit more in detail, you can even see vesicles that fuse to the T bars. So these are the presynaptic terminals. And then you can see here also the postsynaptic partners. And at that time, it was not possible to do that in an automated or semi-automated segmentation way. So at that time, everything was done manually. And that means you need half a year of training to get the 3D vision, and the quality was not so nice, of course. There are some gaps in between, so we don't have every section. And then you have to train half a year, and then with these neurons, these are somehow complex, but not too complex. It takes about 2 days and 2,500 clicks to reconstruct a neuron manually. And for the learning and memory circuit, what Kati Eichler did in my lab, it's about 400 neurons. And now you can do the maths and the calculations, how long it takes. And now you can multiply it by 10,000. And therefore, maybe it took 10 years. Nowadays, it's the quality of the scan is better, and you can team up with companies, and also Google is involved, and other companies that you can invest maybe 20,000 euro, and then you can do an automated, semi-automated segmentation. But you still need experts to have then a look at the reconstruction and match the biology then with what you got from the company. Yeah, but it's much faster. I mean, now you can do this, the reconstruction you can do in 2 months or 1 month with a small group of people. So things are increasing and increasing constantly. Okay, so what we have right now, this is for the Connectome, for the Drosophila larva. In the 2 hemispheres, there are 2,500 neurons roughly, and they give rise to about 730,000 synapses. But of course, there's also the ventral nerve cord, which is still missing and reconstructed in huge parts, but not completely. There, of course, you have more than 7,000 neurons in addition, and a similar number, of course, in synapses. So you see, although you think it's a very simple brain, this is not simple. No, not at all. And now, if you take if you take the entire circuit, 400 neurons for learning and memory, and if you look at the numbers, what is the cable length, what is the number of synapses, then this entire circuit is as complex as one complex neurons in the adult brain, like the APL neuron. And now you can come up again, and you have thousands of neurons in the adult fly brain, and now this is maybe as complex as one neuron in the honeybee brain. And then you have thousands and thousands of neurons in the honeybee brain, and this is maybe as complex as a mouse brain, a single neuron in the mouse brain. And then somehow you end up at the end of this, at the chain, at the human brain or other higher vertebrates. So you see, it's how much it is, how complex it is, and how much data it is also that we can only partially handle with the current techniques right now. Okay, so the basic organization, and that was another thing that we learned already. How do we look at how does the brain work, and how can we look at it? And basically what we now do, at the beginning, the idea was that we just have a single neuron, and to identify the pathway, we look at all downstream partners, and from these neurons we look at all downstream partners, and from these neurons we look at all downstream partners. And then we realized after just 4 of these hops, you basically have all neurons of the brain. So that's not working. So what is working, I think, and what we are still testing, is that you clearly define your sensory input or neurons where you start in the circuit. And then you also clearly identify the output neurons. So what is the circuit, what kind of output you're looking at, and what kind of input. And then you try to reconstruct the shortest way, which is usually only 2 or 3 hops. And then in addition, you can also, if you say you have 2 interneurons here in between, then you can see, okay, what happens if we take 3 interneurons? What is then a circuit that is one hop longer. And by that, you can then compare and get an idea what is the shortest cut, what is the information flow that is a bit more complex, and so on. But otherwise, if you just go on and try to identify something, you're lost. And for the larvae right now, and this is maybe an advantage, maybe due to the limitation also disadvantage, you can define, of course, the visual input. These are just 29 input neurons. Olfactory input, these are just 42 input neurons. And the output basically right now is subdivided into hormones, so secretion of things that modulate the animal or the state of the animal or other things, and two kinds of motor outputs. One is in the SEZ, this is the center all involved in feeding, and the other one is locomotion. And basically, for the first two larval stages, I think that's basically what the animal nearly does all of the 80 to 85% or 90. So within these 5 or 6 days, the volume of the larvae increases by the factor 250. So if you now look at your neighbor and you think next week you meet again, and so you see how dramatic this, yeah. So it's just unbelievable what the larvae, about the metabolism and other things, how this is regulated to get such a massive increase. Because this is really the feeding stage of the animal. Okay, but, and that's a little bit of genetics, so of course that's the power of the Drosophila that we now have established also with Genelia and other people, tools that we can manipulate, can create animals, hundreds, thousands of transgenic animals, where we can manipulate parts of the brain, like the memory center, you show here again, or even single neurons. And the idea is that in the circuit of the brain, of the central hemispheres with the 2,500 neurons, right now we have tools, I would say, to manipulate about 800 neurons individually or 2 neurons at the same time. So, and by that we can, in the circuit, then turn on, turn off with genetic tricks, so that we have thousands and thousands of these tools in addition and can now really work with that on different levels. Yeah. But I promise to not go into the details with the genetics. So, therefore, I will tell you something else. I will show you what we now do. And, of course, the brain is an important part of an animal, but it's not everything. And if you look at the animal, then there's much more. So this is about, so what we did, again, in collaboration with Michael Pankratz in Bonn and Albert Cardona, who is now in Cambridge. So we have now a volume for the entire larvae. So we can reconstruct the entire larvae. And you see, this is just the brain here in this section. There's also lots of gut and the outer surface. If you focus in, there are sensory systems here, there are also sensory parts, there are muscles, and so on. And this is the reconstruction that we started. And here we can follow, of course, the information. Then we have the pharynx, and this is, you see the brain is massive. That's the size in a L1 larvae. But we have all other organs. And now we can also reconstruct the peripheral nervous system, the gut, everything, and look at the interaction of the gut and the brain, of the peripheral system, so that we have not only the information flow that we can reconstruct in the brain, but of the entire animal. Yeah. And just to, does it work? No. Yeah, just to show you also the data. Yeah, so here you don't see that much, but this is really the sections of the entire animal. The resolution is not as high as in the brain, so that's the part of the brain. But a big advantage is now that we can really look at, for example, here at the head region, can zoom in 3D, can look at it, and then we see here, this is a ganglion of the terminal organ here, where you see the cell bodies, you see the sensilla here to the periphery. You can reconstruct it, and you can reconstruct the entire nerve back to the brain. And then we can link that with the connectome in the brain. So we know where the sensory neurons come, and we can link that to the connectome. And by that, we can reconstruct now the information, where does it come, and how is it then processed in the brain. So this is another sensory organ, the dorsal organ. And that is the work of one student in the lab, of Vincent, who has now reconstructed the entire peripheral nervous system. So we know now about every single sensory neuron for this one animal, and have completely reconstructed. We have reconstructed also some body segments with the muscles together with Michael Pankratz in Bonn, and know also the sensory system now on the muscle, and also the motor neurons that innervate the muscles, and then can trace that back with the neurons, also in light microscopy, to the brain, and then see how this interaction works. And with that, we get a complete map now for the entire animal. So from brains to the entire animal. And again, we can use then specific genetic tools to manipulate also the peripheral nervous system and see what happens if you change the sensory system. Yeah.
The staining method for the organs, is this the same approach as for the brain, or is there optimizations one can do for different type of tissue?
So the method is the same as done for the brain, and this is also the problem. Because again, the quality is not so now there are two problems. One is the staining method, and the other thing is that you have to get the fixative into your probe, and therefore you have to make cuts. And whenever you cut the animal, you cut a nerve or an organ or something important you're looking at. So these are the two problems with the volume right now. So there's definitely a lot of room for improvement, and you can really improve or you can modify your staining method if you want to look a little bit more at muscles, if you want to look a little bit more at neurons or some structures and so on. And there are 4 or 5 or 6 different protocols. And depending what you're interested, you can use then different ones. And on the other hand, you have to be careful, because with different techniques, you visualize different parts of the animal in a different way. And it's not so clear what is the real or the most natural representation. Right. But okay, for the first part, I showed you that the central and now also the peripheral nervous system we have described at a single cell level, and most of these synaptic connections are now available. So the circuit is available, the network is available, and then we can now use that and to look at the function of this network due to the accessibility with genetic tools and other tools. Okay. So one example is the dopaminergic neuron. I mean, that is what we did for the last 10 years, and how we analyze the function of the dopaminergic neuron is by looking at learning and memory. Usually what we do is a very, very simple assay where we have a Petri dish with sugar inside, mixed into agarose, which is at the bottom, and we can apply a certain odor. And then we move the larvae, about 30 larvae, so this is a group assay, to another plate where there's no sugar and only agarose, but another odor. And then afterwards we repeat that 3 times, and afterwards we let the larvae choose on a sugar-free plate if they go to the sugar to the odor that indicates a sugar reward or a positive situation, or to the other odor that indicates basically nothing. Agarose has no meaning to it. And if we do that in this way, you see that most of the larvae, not all, but most of the larvae go to that side or prefer this odor. And if we do the reciprocal training, pairing the other odor with sugar, then they go to the other side. And from these 2 experiments, we then calculate one learning index. We do this always in these 2 ways, because it's possible that one odor is more positive to them as the other one. So also these olfactory informations, they have some meaning to the larvae. And it's possible that you have a strong preference already for this one, and then it's not learning, but you would cancel that out in the second experiment, and therefore you do these 2. Okay, and what we see is that basically before the training, the larvae distribute randomly in the ideal case if the odors are if they do not prefer one over the other, maybe here this one a little bit more. But then after the training, it's clearly what you see that in that case, this odor was paired with the sugar in the training, and in that situation, the other one. So the larvae and the three situations are exactly the same. It's the two different odors on an agarose plate. But the larvae change their behavior due to experience. Here naive ones, and here trained in one way, and here trained in the other way. So just by the experience, they change the behavior, and that's a clear definition for learning. And memory if this behavior persists over hours. Right, and usually we and others have now established lots of these protocols and different kind of protocols, but what we're most often working with is salt learning for aversive, high salt learning for aversive training, and odor-sugar pairings for appetitive training. And so what we did now, we went back into the circuit and we looked where are the dopaminergic neurons. And this looks pretty complex, as I showed you before, but here we found out that this is the memory center where the olfactory information and other information comes together, and the neurons that signal if something is positive or negative, if signals the information, a teaching signal, directly into the memory system, these are here dopaminergic neurons. There are 30 in total of these input neurons here, 15 on each side, and from these 15, only 8 are dopaminergic. So there's also other input. We don't know what kind, octopamine and maybe something else, but we don't know. But these are, most of them is dopaminergic input. What you also see, and what is now, if you look at the entire brain, and you see this in this winding paper, the dopaminergic neurons, they get lots and lots of feedback. These are the neurons that get most feedback from all neurons in the brain. And that's maybe also a reason, if this is the same also in humans, why these neurons maybe die a little bit earlier than others, because they constantly process information and based on the connectivity. That's what we can see in the larvae. Okay, and so, yeah, and also in humans, dopamine signaled somehow, that was the latest Wikipedia stuff, that it signals somehow that you go towards a cue or you go away from it. And for years that was a bit a problem, because in flies the idea was that it's only to signal that you go away from something. But in the last 5 to 10 years, people showed also that you see the same, that you have both kinds of informations in the dopaminergic. Okay, the larval system, if you look at these dopaminergic neurons, you have individual neurons that project onto this mushroom body, onto the memory system, which is here, the memory, learning and memory system here, and innervate individual parts. And one neuron is here, for example, the next one would be here, the next one here, so they really tile up the mushroom body in different areas. What you can see here also, there's one, there's another one, and the third one would be here, a fourth one would be here, so they really form sub-compartments. And we looked at that quite some years ago, and when we block the function of these dopaminergic neurons, then we saw at that time the aversive learning is gone. So they are involved in something, signaling something bad. But at the same time, we also saw that also the appetitive learning, the sugar reward learning, was also gone. So both. At that time it was surprising, so we had quite some hard time to publish that. But now it's generally accepted that it's like that. And it took us more than 10 years to understand why and how is that working. And the system is very simple in principle, because you have 4 dopaminergic neurons that innervate these compartments, and they signal the aversive information. And you have 4 dopaminergic neurons, again from another cluster, innervating another part of the mushroom body, and they signal the positive information. So really 4 and 4 neurons, very simple. And if you don't give the larvae a real positive or negative cue, but if you just activate these neurons, then if you pair an odor with the activation of these neurons, then you get positive learning scores for these 4 neurons, the PAM cluster. And if you activate the other 4, then you get an aversive memory. So this is clear. So you really see a separation of the memory system into 2 different parts. These are the ones here, the reward information enters the memory circuit, and here the aversive, the punishment signal, the aversive signals. And this is the information flow, again, what we now know from the connectome and also from adults. So these 2 systems also are connected with each other. That's another thing we see from the connectome. There are many of these feed-forward and feedback loops to keep the system into a balance. And right now, for example, what we also do is we now constantly activate mildly these neurons to shift the balance over 5 to 6 days, and then see if we can induce something like the same you would do with drugs, for example, if you would constantly apply drugs, and if you see the same effects also with the larvae. And first results are really promising. Okay, and the next question, and the last one for this part, is we also wanted to know how different are these neurons of these DL1 clusters. So do these neurons all signal the same information? I mean, this is very unlikely, but was never really shown before. And therefore, we went back to the connectome. And what we saw was not surprising, but what now we had for the first time data that if we start again with the sensory system and try to link the sensory system with these dopaminergic neurons, so from the sensory system to the dopaminergic neuron, this is the sensory neuron, here this is one interneuron, and this is the dopaminergic neuron. This is the CDGF, and you see the same here. And then what we saw is that we have one neuron, the C1 neuron, and this is not really getting sensory information or that much sensory information. However, we have another one, and that gets really a lot of different types of sensory information. I mean, this is color-coded, but I will not go into detail. So, and then you also have something in between. You have 2 neurons that get some information in between. The other surprising thing is that each neuron gets a different input. However, not completely different. Some things are overlapping. So you see these neurons, 1L and 1,3. These are 2 different interneurons, and they are also here, and they are also here. So partially it's overlapping, but it's also partially different. And you have something like more like a generalist and something like a specialist, or if you look a little bit more into that, of course, this neuron does not only get sensory information, but also from the brain a lot of feedback over other neurons not connected directly with the sensory system. So this neuron is more or less involved from the connectome only, in processing information, feedback or processed information in the brain already, and not directly to the sensory system. Okay, and oops, that's weird. And yeah, we also see that if you look at the function of these neurons, we see that also then we see that also if we analyze the behavior, what happens if we either kill or if we activate these neurons, then we see that they do also partially something different. So if we kill, for example, this neurons, this is the C1 neuron, then we see no effect on the learning. However, if we kill this neuron, the G neuron, the generalist, then we see a reduction. So and for the activation, if we activate this neuron or that neuron, in both cases we see an aversive memory. So you see also somehow the function, we can link it now with the connectome, and it makes somehow sense. Not 100%, of course it's more complex, but somehow. Okay. So yeah, so basically you have the dopaminergic neuron signal positive and negative teaching signals. We have these 2 clusters of 4 neurons each, the PAM and the DL1, positive, negative. They get no directly sensory input, but usually over one interneuron. So they are not completely directly connected to the sensory neurons. and you also see some feedback and other stuff of processed information. And the information they get is only partially overlapping, and each one gets a specific set of information. Okay, and yeah, with that I want to make a cut now about something different. So usually when we work with optogenetics, with these genetic tricks and activate neurons, then what we can do is, of course, in this case, for example, you take a line where you know what happens. In this case, we activate all motor neurons with this new generation of blue light sensitive tools. And this is what you can see here. And now if you activate the blue light, it's so fast, you can't see it clearly, but you see basically no movement anymore. So within some seconds, and then you turn off the light and they start crawling again. So these new generation of tools are really so specific, and we only use 1% intensity. In former times, we had to use 60–70% of power of these LEDs. So we can really turn on and turn off these neurons within seconds in the living animal. And of course, what we then did, we expressed these generodopsin XXL, this optogenetic tool, in a lot of different driver lines, and just to see what happens if we change the behavior. Can we see a behavior change? And in one line, we saw something like this. And at the beginning, we thought, so this is already with the blue light activation. Somehow is different. Something is different, we thought. And what we saw is that they really group together. So this is, if you compare it to these larvae, Y-type larvae or other, if you activate other neurons or other cells, then you don't see these grouping behavior. So really the animals came together. And we looked a little bit more in detail. If you activate here in this combination, if you activate, then we saw they group together. But if you look at these ones or here, so it's not that they really do something. It looks like a kiss, but it's not a kiss. So they really try to cannibalize each other. And if you let that run for longer time periods, then they really kill each other. So we quantified that, and to our big surprise, we really can, in that case, so this is the clustering behavior, so they don't form clusters of lots of animals, so they usually are separated. But when we turn on here we did it with a red light, with another genetic tool as an additional control. But if you turn on these neurons or these cells, then they start to cluster and they start to cannibalize each other. And then you turn off the light and you see within seconds they stop with the behavior. So we have really a trigger to induce clustering and also cannibalism. That's the next slide. So the question was, what neuron is this? And it's also a long story. We mixed up some lines and so on and did the molecular stuff again. And we found out it's called lily Gal4. And this is somehow in connection with the foraging gene. There's also a long story, and it's not super clean with the foraging stuff. So this is called lily Gal4. And we looked at the expression. So we found out that you have nice expression here in the antenna lobe, but these are inhibitory interneurons, which is not so straightforward. You have something in the last segment, but that differs also between female and male larvae. So we see the behavior in all larvae. So that's also not very promising. And then you see something here, which is pars intercerebralis, which is maybe insulin-like peptide, which would also somehow fit. But the expression is very weak, and same as here, also randomly, somehow randomly. So what we saw, however, is usually if you look at the entire larvae, you wouldn't see anything here in the brain because the salivary glands are lighting up as hell. So So these are really the glands, and therefore we were searching in the literature for other lines that specifically label the glands. It looks a little bit different. However, it also labels these. There are not too many cells in the glands. These are huge cells, and it's a monolayer basically, and secreting lots of stuff. And there's not too much known about the glands. So it's known that they are located here. They can be pretty huge, and then they form a pore here, and then secrete as material or proteins or other stuff into the foregut or the pharynx. And also it's known that they can secrete stuff into the hemolymph and thereby regulate also the growth of the animal. So that's the glands. And at the end of larval life, they completely switch and produce only glue, 7 different glue proteins. And with that, this is then put over the entire larvae to form the pupae during metamorphosis, or in the beginning of metamorphosis, which is also quite interesting behavior. Yeah, so that's, and we repeat it then with this gland-specific tool, and we see more or less the same. So we can trigger this, not maybe at a little bit lower level, but we can clearly trigger the same behavior. So they come together, and they also start to cannibalize each other. So with this optogenetic tool, we also tested about the feeding. So do they eat more if we put them on a petri dish with agarose with red dye? And we clearly see that they eat more than the controls. And also very surprising is if we use a tool to inhibit to silence these cells. I mean, these are not neurons, these are cells. Then within 3 hours, the larvae die. Right, and this is not due to dehydration, because if you do this in water, you see the same effect. Yeah, so that's the effect where we are right now. So it's known from different studies that larvae usually tend to dig into the food together in groups, that this is some kind of behavior where they come together. It's not clear what kind of substance or what is the mechanism or why we see that behavior, but we can really trigger that, turn it on and turn it off specifically. Yeah, and that's the last part. So we can induce optogenetically the clustering behavior with different genetic tools. The behavior change is really acute. We can really turn it on and turn it off again within seconds. It triggers a really dramatic increase in feeding. And now we are working with several techniques. We try to identify the nature of the signal. But we are at the beginning of this to identify that. Okay, and with that, I would like to thank you all for the attention, and especially I would like all people here, but also Denise and Vincent. Vincent is doing, as I said, the EM reconstruction of the peripheral nervous system, and Denise is doing all the stuff on the dopaminergic system. And yeah, thanks a lot, and I'm more than happy to take your questions. Yeah.
Okay, thanks a lot. And we have questions already? Let's start at the beginning.
Yeah, thank you for this super fascinating talk. I would like to ask something about the something else in the end. And about the antennal lobes, you said it's likely, the expression that you also found was in these interneurons. And I wonder, is there any possibility that what might actually also change is how the other larvae smell to the now cannibalistic larvae, that all of a sudden the smell of the other larvae is appetitive to them?
Yeah, I mean, it's known there are different pheromones and so on, and in different ways. But if you look at these neurons here, I would not really exclude it, because also the effect that we see with Liligal 4 is stronger than the effect what we see with only if we do it with the glands. And it can be that it's a mix of, I mean, there's also some but this is very, very weak here in the gustatory part. This is the olfactory part. This is insulin. So maybe it would somehow fit that several systems are teaming up to get this dramatic change in the behavior, and plus the glands in addition. Other body parts are not involved, but it's also possible. I mean, you can, with these are the GABAergic, 2 or 3 GABAergic interneurons, and with that, of course, you can then, if you activate it, you can downregulate some olfactory input. And if that works as a filter, then maybe other cues can take over. It's possible, yeah.
Thanks. That was really, really interesting. So I have another so actually I had very similar question, but a little bit differently pointed. So, and this is following a new recent paper from Vienna looking at cannibalism in locusts, and they actually found that the males are producing a pheromone that to prevent others to cannibalize on them. Yeah. And then if you block the ability to smell that pheromone, you could stop the cannibalistic interactions. And the question whether orco flies, whether you could deactivate the cannibalism by also adding an orco on top, so that they cannot smell. They cannot. Yeah.
As I said, I'm not sure if this is. I mean, you see the cannibalism in the end, and I'm not even sure if olfaction is involved. It can well be that you secrete some stuff, and then this secretion triggers. Or that even it's a mechanosensory feedback, that you secrete something into your front gut, and therefore this induces some mechanosensory feedback, and you have to then chew and triggers the motor neurons there. There are also sensory neurons in the front gut that we know now. But on the other hand, also a similar mechanism we identified also and published for the eggs of the larvae. So usually there are some pheromones on the eggs, and if the eggs and that somehow shields these eggs from cannibalism. And if you then destroy artificially, or if you bleach the eggs a little bit, then you remove this layer, and then the larvae start to cannibalize. And also, larvae from different species can somehow sense that or not sense it, and therefore you see cannibalism in different species. And on an apple, a rotting apple, for example, you have different spots of different species of Drosophila, so they do not really mix, so they somehow separate each other, separate from each other. Yeah.
Okay.
Cool.
Yeah, very simple question from a computer scientist. I mean, you said like they eat so much that they get like 250 times bigger within a week. So I would say it needs a very strong signal not to eat something. Yeah, so it's just like maybe it's just like something that is missing, and, and as a matter of fact, they just start to eat whatever is in front of their mouth. Yeah.
Yeah. I mean, we don't have the data, but that's also what we see. I mean, if we try to starve the animals, the larvae, it's not really working. So also for the memory in adults, the positive memory works only if you starve the animals, but larvae, you don't have to starve them. So and I don't know if, there are some effects. We also have effects. And yes, true, but for the memory, somehow. And but it's also possible that their metabolism rate is so high that they constantly have to support, and also it's not maybe possible then to invest this energy into long-term memory, only into short-term memory, and because you need so much energy all the time and for the metabolism. There was one more.
So we have one question in the Zoom.
But there was one at the back also.
And I have to run, but it's actually also the question I had.
Okay, then.
So Ian is asking, are the larvae actively aggregating, or does the aggregation result from the fact that when they contact others, they begin to cannibalize and thus stay in close proximity? So it's basically they think they find food and just stay, so it's not really that they want to...
Yes, both is possible. I think rather the last thing is more likely, because they really attack each other, and it's not that they are completely trapped, but they really, as I mean, they also show some response to plastic, but to larvae the response is higher. That's also what we saw. It's not that, but it's not that they secrete something, but they just need they get some information, and then they start with the cannibalism behavior or increase even the mouth hook, the rate with how they contract their mouth hooks, and then by that I think they are aggregating because of this. But we have to look a little bit more careful into that. Yeah.
Hi, so I have a question follow up that. So I still don't understand what's the definition of clusterings and cannibalism, and have you ever tested to what extent this cannibalism behavior is specific to the same species or other type of animals? Yeah.
I mean, the clustering is just the number of larvae that come together over time and really are in contact with each other. So that's how we define the clustering.
So an actual physical contact?
Yes, okay. Yes. I mean, we are now also thinking to define certain areas and ROIs, but now it's really the physical contact that we define here. And we only tested larvae. However, what we also did, we tested larvae of wild-type larvae and these ones to see if for example, they show a difference, if they would secrete something onto mark this larvae, or if they also would secrete something onto the agarose, a signal, but that is not the case. And this is also shown here, so we can immediately turn it off again. So it's not that I don't think that they secrete something or mark the surface of another larvae or of the petri dish with something. It's another information they use. Yeah.
Have you also tested the activation on a single individual and see what happens at the individual level and see if the clustering is somewhat a consequence of the presence of the individuals or just an activation of the motor system, or have you, I don't know. No, we haven't. I mean, usually we do these in groups of 20 or 15 to 30 larvae or up to 20 larvae, and we never looked at individual ones. True, we should also look at single ones and see what happens. Yeah, more in detail and check what changes for individual animals. Yeah, but so far, no.
Are there any more questions? Yeah.
Yeah, thanks for this, especially the something different. This is all great. I was actually wondering one step before in your olfactory learning paradigm. I was wondering if you'd given any attention to the sort of 30% of individuals that don't seem to learn. Is there anything functionally different about them?
Yeah. So when you take these 30% that the non-learners and separate them from the learners into two groups and you retest them again, then you see again that 30, 70, 60% show the behavior and 30% not for both groups. So I think it's not really that you can in each experiment see the learners and non-learners, two different groups, but it's just based on the experiment that in some cases it works and in others not. And yes, yeah, right. And that's also shown that was also shown for adult flies. Yeah.
Okay, more questions? No, also in the chat, I think. No. Good. Then thanks a lot, Andy.
Thanks.
And thank you all, and I wish you all happy holidays and see you next year.